cell therapy, it's much more modular because, when we design a car, the car has several different components. And if we change any component, we have to open a new IND, and that takes a long time. So there's now the FDA has a system set up that will potentially let you do kind of an umbrella protocol where you still need to submit all of the required data for each of your variations on the theme. So you might have a CAR recognizing a certain antigen, and then you might have the same CAR with some kind of armament, or armament A, armament B, and you might be able to study them all in one clinical protocol under kind of a nested IND. So that would be very, that would certainly optimize our approach here, because otherwise it's very laborious to obtain an entirely new IND for a very similar therapy. And that problem really hasn't existed before, because of course the regulatory structure we have is largely developed in response to small molecule and then more lately monoclonal antibody therapy. And in those cases, it wasn't generally, like if you change even a single atom on a small molecule, you can end up with a very different small molecule. So it wasn't modular in the way that cell therapy and even if you don't change the CAR, changing the manufacturing protocol, and there's some innovation there. So that traditional CAR-T autologous manufacturing protocol is 9 to 11 days. Novartis is now publishing some, showing some data at conferences from its T-Charge platform, which is only 2 days. And UPenn recently published on a 1-day protocol. So those would have quite a large impact on because the process is the product, pretty much, for a living cell. So even if you don't change anything genetically about your therapy, changing the process can have a big impact. And being able to do that efficiently without having to undergo the full IND process for every incremental change, I think, would be important.